Yeah, I think I got the answer. Doesn't have fun, yes.
Nice watch.
The two of us, not just go.
It's the same thing as except very unlikely they will consider our.
I'm not sure.
Maybe it might go.
So she said.
Hi everyone, welcome to the Industrial Talent Seminar. It's a great pleasure to introduce Dr. Michelle Mastrianni. She's an All-Rise Fellow at the FDA. She graduated from the University of Minnesota about a year ago. Her PhD advisor was Professor Dimitri Bilik, who is sitting over here. And she's going to talk about regulatory science challenges for AI imaging devices.
Hi, everyone. Thank you so much for the invitation, Gilad. Thanks for having me and coming, everyone. So as Gilad mentioned, I graduated in December last year. And since January, I've been working as basically a research postdoc at the FDA. I'll explain what all these acronyms mean, but I work in the Division of Medical Imaging, and I'm looking forward to kind of sharing the goals and research priorities of our division. So the first part of the talk will just be introducing these acronyms. How does my division, DIDSR, fit into the FDA at large? And after that, I'm going to discuss a few examples of research projects within the division, and then I'll talk specifically about two projects that I've been working on for the whole year. These are very probability and statistics based. With imaging, you may expect me to talk a lot about Fourier analysis, and actually these projects are more on the probability side. But there are also really interesting applications of Fourier analysis to imaging in the division as well. But yeah, so I'm looking forward to sharing my specific projects, and then at the end I'll touch on some opportunities with the FDA that students may be interested in. So most people, I think, know that the FDA stands for the Food and Drug Administration, and as per its name, deals with the regulation of food and drugs in the United States. And indeed, the Center for Drug Evaluation and Research is the largest center at the FDA. We also have the Center for Biologics Evaluation and Research, which deals with vaccines and genetic therapies. And then, of course, the Center for Food Safety. But the center that I work in is the Center for Devices and Radiological Health. And this is all about the regulation of medical devices. So some people don't know that that's a big part of the FDA as well, is device regulation. So the Center for Devices and Radiological Health is a team of over 1,900 public health employees with expertise in different subject matters. And the center is kind of, or full-time employees at the center sort of split their time between two things: regulatory work and research. And sometimes they inform each other. So the full-time employees at CDRH provide subject matter expertise to the review of medical device submissions. And then with the rest of their time, they conduct research, creating tools and software to help companies in those submissions, and also guidance documents to support the review of medical devices. And as of recently, last month actually, we have a new center director. So our previous director, Jeff Shurin, was the director for 15 years. And now we have Michelle Tarver, who is an epidemiologist and ophthalmologist by training. So we're all looking forward to her leadership at CDRH. And this graphic gives you an idea of how large the center is. So as I mentioned, about 1,900 employees, and we get about 22,000 pre-market submissions from companies a year. This number actually might be out of date by now because this number is just growing rapidly every month. And even anecdotally, my colleagues who do regulatory consults with companies are getting more and more consults every single month. So this might even be out of date. Within the Center for devices in radiological health, there are several offices. Some of those are more administrative, some are more policy and regulation focused. But our division is within the Office of Science and Engineering Laboratories, which is the part of CDRH dedicated to regulatory science research. So it's a very research-heavy office. And there are about 180 federal employees and up to 180 visiting scientists, whether that be postdocs or scientists visiting from universities and so on. And yeah, within OCEL, this is about the number of consults, although again, that may be out of date by now. And then my division is, we call DIDSR. It's the Division of Imaging, Diagnostics, and Software Reliability. And it's the part of OCEL dedicated to imaging research. So the other offices within OCEL are, I believe, the Division of Applied Mechanics, the Division of Biomedical Physics, and the Division of Biology, Chemistry, and Material Science. And all of those divisions, their research supports understanding the interaction of medical devices with the human body, which is obviously a very important area of research. But by contrast, DIDSR, most of the work in DIDSR is in evaluating medical imaging devices. And these are sort of the four main research programs within DIDSR, and some of those overlap with other divisions in OCEL. So we have about 45 federal employees and about as many fellows and students. We're also hiring more. That'll be one of my spiels at the end here. But yeah, just again, to give you an idea of how many consults DIDSR sees every year. So before I touch on or get kind of in the weeds with my own research projects, I just want to give some examples of research projects within DIDSR and also emphasize how the rise of AI, ML in medical devices has impacted the regulatory work that we do. So even from this graphic, you can see there's, you know, a sharp increase in the last 5 to 10 years of AI, ML enabled medical devices, particularly in radiology for diagnostic purposes, but also for patient management and monitoring in, for example, the cardiovascular and neurology spaces. And because we're in Minnesota, for those of you who don't know, I just want to point out that Minnesota has been a big hub for cardiovascular devices. The first pacemaker was invented at the University of Minnesota, and the cardiovascular device space is still really big here. But yeah, most of these are in radiology, but as you can see, there's kind of a sharp uptick in the last 5 years in these types of devices. And with those device submissions come a lot of questions about how best to evaluate them, what are the statistical methodologies needed, et cetera. And there are several interrelated topics to consider, such as data acquisition and data quality, study design, AI algorithm design, which can often feel like a black box in some cases, and also intended use. There are many different types of intended use for devices, and I'll get into a couple distinctions of that later. And then generalization and bias. So how well does an AI algorithm generate to or generalize to other populations or over time, and different types of data and all of that stuff. Okay, so I mentioned kind of the 4 areas of research within DIDSR. One of these is medical imaging and diagnostics, and these are just a few projects in that area that are ongoing. One down here, you see evaluating skin color effects on pulse oximetry. So optical devices take pulse oximetry measurements to measure blood oxygen, and there's a lot of research that skin color can affect the accuracy of these measurements. So this project is all about kind of developing metrics and methodologies to assess the quality of those measurements and assess uncertainty and bias. Looking at data drift and bias, as I discussed on the previous slide, so monitoring AI kind of over time, looking for ways to measure the difference in distributions month to month, for example, of AI performance. Evaluating imaging systems with photon counting detectors, so in spectral CT, coming up with ways to evaluate image quality of spectral CT systems that employ photon counting detectors for tissue contrast and enhanced image quality. And then AI/ML has some overlap with medical imaging and diagnostics on the previous slide. One area that's growing that you might not be surprised to see is evaluating large language models, particularly in the generation of radiology reports. So I've learned a lot about radiologists and their jobs this year, and something that I've learned is generating reports after reading an image. This is the part of the job that radiologists dislike the most and really would like AI to help with. But of course, such LLMs need to be evaluated for their accuracy and consistency. So this is an ongoing project in the division. Also, uncertainty quantification. So uncertainty in AI/ML enabled devices can come from data acquisition, kind of different types of data. And there's a lot of places where you could have uncertainty in AI outputs. So uncertainty quantification is also a big area. And then on the right, assessing in silico models. So in silico models are simulated models that that are based on experimental or clinical data, and also research. So one of the big projects in DITSER is coming up with assessment techniques for these in silico models. So how well do they perform, particularly for pediatric applications where there's often a lack of real clinical data for a variety of reasons. There can be a lack of pediatric data. And then my project on the left here, which I'll talk about more in depth, is developing statistical methodology for the evaluation of AI triage and rule out devices, specifically in mammography, but also other modalities. And yeah, we'll get into more of that later. And then some other research areas, digital pathology is growing a lot. So digital pathology is the process of converting traditional pathology slides, which are glass slides with tissue samples, into digital images for easier analysis and also integration with AI. And one of the projects that's going on there is creating a large validation data set established by pathologist annotations. So the goal is to have this public data set for use by companies that's really good quality data that's been validated by pathologists, by a number of pathologists, which companies can use in their research. And then we also have a small group working on augmented and virtual reality projects as well. Okay, so those are several of the projects going on in DITSER as a whole. I'm going to discuss two of my projects in more detail now. And like I said, they're fairly statistics and probability heavy. So the first project that I'm just going to discuss is developing statistical methodology for evaluating rule out devices, and I'll tell you what that means on the next slide. But for those of you in the room who are not familiar with ROC curves, I just want to briefly go over what an ROC curve is, because this is really fundamental in diagnostics. So let's say that we have some diagnostic. This could be a radiologist, like a human radiologist, or an AI device. And we're able to model their output scores. So maybe they're outputting scores that represent kind of confidence in whether or not an image is cancerous. And so let's say we can model those output scores via these normal distributions, where the distribution on the left is for true non-diseased patients, and the distribution on the right is for true diseased patients. And let's just take, for example, this red line as one possible decision threshold. So that threshold could mean all images with a score higher than that threshold are deemed cancerous. All images with a score lower than that threshold are deemed non-cancerous. Then the false positive fraction at that threshold is the area under the curve, under the non-diseased curve to the right of that threshold, because that represents the number of, or the proportion of patients falsely called positive by the diagnostic. And similarly, the true positive fraction at the same threshold is the area under this distribution on the right, which is the disease distribution. And this is the proportion of patients that are called positive correctly by the diagnostic. And you can understand these false positive fractions and true positive fractions as follows. So this is the number of false positives divided by the number of true non-diseased cases. And the true positive fraction is the number of true positives divided by the number of true diseased cases. And so at this red threshold, we can plot the FPF and TPF up there, corresponding to the red point. And maybe at the yellow threshold, we do the same thing. And we do that for every threshold. So the thresholds parameterize this ROC curve. And something to note is kind of the better performing the diagnostic is, the higher the area under this ROC curve will be. So we refer to that as the AUC, the area under the curve. And in general, the better the diagnostic, the higher that AUC. So I mentioned rule out devices, but I haven't said anything about what this means. A rule out AI device is intended to autonomously remove AI negative patients from radiologist review. So this is just a little graphic explaining how this may work. Maybe an AI outputs scores from 0 to 1 and the rule out threshold is 0.2. Then every image with a score less than that threshold is deemed automatically negative by the AI device and not seen by a radiologist. And this table on the left kind of represents the effects of use of a rule out AI on diagnostic accuracy. So if the radiologist and AI agree, there's really no effect for that particular image. If the rule out AI gives a true negative to an image that would have been called a false positive by a radiologist, that's a benefit to the diagnostic accuracy. And then vice versa, if the rule out AI calls the patient a false negative, which is really the risk of this rule out scenario, but a radiologist may have spotted a true positive, that's a loss to the diagnostic accuracy. This table doesn't fully capture all the benefits and losses, because there's also a benefit to radiologist workload. If they can spend more time on more complicated cases, that's another benefit to a rule out device. So there are a lot of things to consider, but we're interested specifically in the diagnostic accuracy. And also what this table demonstrates is that the radiologist AI correlations will affect the combined scenario compared to the radiologist alone scenario. And there are several papers looking at metrics for evaluation of rule out devices that do not account for these correlations. So really nowhere in the literature are these correlations taken into account. Mammography is a specific example of a modality where a rule out scenario could be attractive, because when you compare to like a lung CT scenario, there are several conditions that may have to be simultaneously ruled out in a lung CT, like pneumothorax, pulmonary embolism, rib fracture, et cetera. All those different conditions would have to be ruled out simultaneously. By contrast, mammography is a bit more of a simple task because you have an AI looking for lesions specifically. And so there's really only one condition that needs to be ruled out in this scenario. So in the US, the standard of care for mammography screening is a reading by a single radiologist, whereas many places in Europe, including Denmark, the standard of care is a double reading. So they have to have 2 radiologists legally look at every image, whereas in the US, only a single radiologist has to look at every image. And because of this double reading, it's been easier to conduct rule-out studies in Europe. So there are several papers coming out of Europe, including this one from earlier this year, that look at rule-out scenarios actually implemented in clinic, where instead of ruling out entirely from radiologist review, you're basically ruling out a certain set of patients deemed high confidence negatives by an AI. So here, maybe the AI outputs scores from 1 to 10, and those with scores from 1 to 5 are deemed high confidence negatives. And those patients go directly to the second radiologist. And then those with higher scores still have to be read by both radiologists. So in this paper, this was a 2-year study, and they implemented the rule-out scenario midway through and found that a lot of metrics like cancer detection rate, false positive rate, radiologist reading workload improved under this rule-out scenario. Like I said, there are a couple papers looking at rule-out theory, so how do we actually evaluate these devices? But they completely ignore the impact of the correlations of radiologist and AI scores. So for example, in this paper, these 2 equations involving the sensitivity and specificity, which I don't think I mentioned earlier, but sensitivity is the true positive rate. Specificity is 1 minus the false positive rate. But the important thing is that both of these equations only hold if you assume complete independence of the radiologist and AI, which in practice is not a reasonable assumption. So our idea was to develop a framework for ROC analysis of rule-out devices, accounting for the radiologist and AI correlations. And one advantage of an ROC curve is that readers may change decision thresholds with the presence of a device. So having a rule-out ROC curve would essentially allow us to move along the curve and estimate radiologist performance at different thresholds. This table gives an example of how radiologist reading or decision thresholds may change with the presence of a rule-out device. It's from the Lauritsen paper from the previous slide. And you can see that the recall rate among patients seen by both radiologists. So in year 1, the AI system was not implemented yet, and so all patients were seen by both radiologists. In year 2, the AI system was implemented. And so you only have these high confidence, or suspicious rather, cases by the AI being read by both radiologists. And the recall rate goes up a lot, which makes sense because among patients seen by both radiologists in year 2, there will be more true diseased patients. But then you also have kind of the impact of the radiologists are aware that these are suspicious cases. So yeah, accounting for those change in decision thresholds, it's really advantageous to use an ROC curve so that we can kind of move along and estimate performance at different thresholds. And the way that we have been doing this modeling is with bivariate copulas, which are essentially ways of creating bivariate distributions with arbitrary marginals and correlations. So as in the univariate case, you might remember that binormal distribution in the univariate case, where the distribution on the left was non-diseased and the distribution on the right was diseased. And as in that scenario, we also need kind of a diseased copula and a non-diseased copula to get the true positive fraction and false positive fraction at each threshold and be able to plot an ROC curve. So for the diseased copula, the inputs will be the diseased distribution from the radiologist, the disease distribution from the AI diagnostic, and then a parameter related to the correlation between the scores, the radiologist and AI scores on the true diseased images. Similarly, we have a non-diseased copula. And then this plot on the right, you can think of these density plots as sort of like a product of the radiologist and AI marginals in some sense, where that product is determined by the copula function itself. There are a couple different examples that I'll go over on the next slide, and then also the correlation. And you can see just from this example that, for example, if we set the AI threshold at 0.5, everything to the right of that threshold is called positive by the AI. And so all of this gray space to the right of that blue threshold will be false positives. But then you also have the radiologist decision threshold. And so with the radiologist decision threshold, you can kind of eliminate this section of false positives. So rule out is really a chance to eliminate a lot of false positives. And there are several different types of copulas that we can use for this modeling. Probably the most popular type of copula is a Gaussian copula, which maps the marginal distribution of each variable to the standard normal, and then combines them under a multivariate normal CDF. And you can see, and Gaussian copulas take as input Pearson's rho, and you can see like for a higher correlation, 0.8 on the right, this copula density plot becomes a lot more concentrated along the diagonal where you have perfect correlation. And then we're also using Archimedean copulas, which they're basically expressed in this form where you have a generator function phi with certain properties. And the correlation measure that kind of plays well with Archimedean copulas is Kendall's tau. I'll discuss that on the next slide. But you can see just from these density plots, the Frank copula is a more moderate copula, while the Gumbel copula is good for modeling upper tail dependence, and the Clayton copula is better for modeling lower tail dependence. So kind of depending on the context of your problem, one may be better suited. Like I mentioned, Gaussian copulas take Pearson's rho as input. This is well suited to the Gaussian copula because both rely on the assumption of linearity. So Pearson's rho is defined based on covariance and standard deviations, which are inherently linear concepts. And then Archimedean copulas are more flexible and capture nonlinear dependencies. So Kendall's tau is better suited because this is a rank-based measure of correlation that relies on ordering rather than actual values of the data. And there's this nice explicit relationship between Kendall's tau and the generator of an Archimedean copula, which I like. Okay, so far I've been talking about rule out diagnostics, but there's also some interest in, for example, combining a rule out diagnostic where maybe you rule out 30% of high confidence negative patients, and then you rule back in some small percentage of highly suspicious patients. So if an AI thinks an image is really suspicious, you would automatically recall that patient. And you could potentially combine these two diagnostics. And rule-in diagnostics are really simple to model in conjunction with rule-out diagnostics. You can think of a rule-out diagnostic as a believe the negative scenario, where if either the AI or radiologist outputs a negative, the patient is deemed negative. And in a rule-in scenario, if either the AI or radiologist outputs positive, the patient is deemed positive. And you have this nice relationship, which I just came up with. I don't think it's in the literature anywhere, but it's one of these things that's just like not difficult to derive, but it's potentially useful. And it's based on just inclusion-exclusion principle. These are just probabilities. So these are really simple to model in conjunction with each other. So I've been talking a lot about correlations and wanting to capture the correlations in these models. And one of our questions was, how do the correlations affect the performance of the diagnostic? And as I mentioned earlier, AUC is really like the main metric that we use to capture how well a diagnostic is performing. The higher the area under the ROC curve, the better the diagnostic. In practice, the AI device, like in a clinic, will be fixed at a specific operating threshold. And so this piece of the copula, which is like the AI marginal, will be a fixed number. And so a consequence of that is that the rule out curve, which, okay, this is a GIF, but you can see at the beginning of the GIF, I'm kind of altering the FPF. So I go from right here, 0.1 to 0.7. And now the rule out curve is at 0.7 and it stops at that operating FPF. So it's a bit of an unconventional rule out curve that doesn't go all the way up to the upper corner. And by contrast, the rule in diagnostic, which is in light green, will start at its operating FPF. But you can see, just based on this GIF, if you pay attention to the correlations up here, that as the diseased correlation goes up, the area under the rule out diagnostic goes up. And as the non-diseased correlation goes down, which we'll do in a second, the area of the rule out AUC goes up. And the converse effects happen on the rule in AUC. And yeah, we were able to prove this for the copulas that we're using with their respective correlation measures, Pearson's row in the Gaussian case or Kendall's tau in the Archimedean case.
To be sure.
Yeah.
So these are, if I look at the whole domain, these are invalid ROC curves because they have to be increasing, right? So you only care about the middle of the domain, or?
So yeah, we only really care about the like going from 0 up to the stopping point FPF. But if you wanted to extend the ROC curve, this rule out curve would really agree with the AI curve past that point. And so you can just extend it by having it be a little bit piecewise. So it's like the rule out piece and then it agrees with the AI curve past that FPF value.
Even, on the left, the light green one is not, I think it's not, yeah, it's kind of a bit strange, so.
Yeah, the light green one is a rule in scenario. And so, so basically the diagnostic can't go below this FPF anyway. Yeah, so they are a bit unconventional, but still the AUC captures something about how well the diagnostic is performing. Or we could use like a partial AUC, so normalized by the range of allowed FPF values. Thanks for the question. Okay, so I want to talk a little bit about some empirical validation that we did for this copula modeling. This data here comes from a reader study conducted at Radboud University in the Netherlands. And basically 12 readers read 150 mammography images, 75 cancer, 75 non-cancer, and assigned a score to each image. And then an AI system read the same images. The performance of the AI was extremely good. So this is the AI ROC curve in purple. The empirical one is the choppy one, and then we fitted a parametric curve by assuming normal distributions. And then the radiologist, the combined radiologist ROC is in red here. And then we got an empirical rule out distribution by basically at some threshold. So over here, we have a rule out FPF of 0.44. And that corresponds in this case to ruling out 30% of the patients. This corresponds to ruling out 20% of the patients. But at that operating threshold, we basically threw out all of those patients that the AI deemed negative and then recalculated the radiologist sensitivity and specificity at each point. So you get this gray curve representing what we project the empirical radiologist performance would be under rule out. And then the blue curves are two copula models, so Gaussian and Clayton respectively, and they agree quite well with the gray empirical rule out curve. And
To be sure.
oh yeah,
So maybe the AI is doing so well, I don't need to. Yeah.
that is a great question. Yeah, that's something we've been talking about a lot. I think, yeah, currently, at least in mammography in the US, currently, it's not possible to just have an AI read all the images. But this suggests that, the AI is doing a lot better than humans and even better than the combined scenario. So it's an interesting, like the AI has gotten so good that, yeah, this is an example where actually the AI alone curve does even better than the combined scenario. So yeah, and I want to emphasize the correlation effects also on these rule out curves. So on the left is the plot from the previous slide where I'm accounting for the true correlations. The middle plot, I'm using an independence assumption where I'm assuming the radiologist and AI are completely independent. And then on the right, I'm assuming that the AI and radiologist are perfectly correlated. And you can see that this overestimates the AUC and this underestimates, or the ROC at every point. And this, yeah, overestimates, underestimates. So the use of the actual correlation measures is really important in this modeling. Yeah, and so for this project, we have sort of concluded a large chunk of it, but there is still some interesting future work. So uncertainty and variance estimates for rule out and rule in AUC, the area under the ROC curves, and also doing subgroup analysis like on patients with dense breasts, for example. And I am likely moving to some other projects within the division. So there is actually some funding to continue this work through 2025. I know it's sort of short notice, but if anyone's interested, please talk to me. Okay, and then this next project is, I think it'll be interesting to this audience. So this is all about evaluating CAD triage devices. So up to now, I've been talking about rule out, but triage devices basically rearrange a patient queue. So in a scenario without a CAD-T device, you basically have a first in, first out scenario where patients are seen by the radiologist according to when they enter the clinic. Occasionally an interrupting case or an emergency case might cut in line. But in a with CAD-T workflow, patients or images would be read by AI, and then those that are deemed as positive by the AI would be put into a higher priority class and jump to the top of the queue. And occasionally you would still have an emergency case cut in line. So companies often claim CAD-T devices prioritize AI positive cases so that likely diseased cases are read and treated earlier. And my mentor this year, Aleem Thompson, this was a big project that she worked on when she was an ORISE fellow. So the first sort of problem that she worked on was just looking at CAD-T with one disease condition. And the overarching question is, can we kind of model or predict waiting time per patient, specifically diseased patients, in a with CAD-T versus without CAD-T scenario, with a number of different input parameters. So you have read times, traffic, disease prevalence, and so on and so forth. So she developed this software called QCAD that basically does just that. It takes these input parameters, traffic density, arrival rate, fraction of interrupting patients, AI performance, disease prevalence, number of radiologists, and reading rates, and then outputs wait time savings and delay estimates in a with CAD-T scenario versus a without CAD-T scenario. The software is nice because it has both simulation and then validation with queuing theory. So she actually used like queuing theory, mathematical theory, to validate her simulation. And all that theory is described in this paper. This is just one of many interesting diagrams in that paper. It's a Markov chain transition diagram for one of the most simple models where you have a without CAD-T scenario and one radiologist. And the busy periods in that scenario are denoted as states where, so this would be the fraction of interrupting patients, or the number of interrupting patients, and this would be the number of normal patients. And when you have interrupting or emergency patients and you have more interrupting patients than radiologists in the clinic, that's considered a busy period. So if this was a true full diagram, this would actually be infinite in the vertical direction as well. But she employs this recursive dimensionality reduction technique to kind of truncate the diagram and just represent all of these as busy periods. And the math is way too much to go into now, but I just want to kind of give you a sense of some of the interesting stuff that goes into this project. So that was for one condition in clinic, but we could look at an expanded problem statement where a patient may undergo a number of modalities, CT, MRI, ultrasound in the same visit. And also you may have AI targeting multiple disease conditions. So yeah, in reality, in clinics, these queues are going to be a lot more complicated than just one AI targeting one disease. And so another part of my time this year has been helping expand the QCAT software to a multi-disease or group scenario in which each condition may or may not have an AI targeting it. So maybe you have different groups of patients where for all AIs in this group, like AI4 and AI5, they're going to read patients getting tested for every disease D, E, and F in that group. So you can imagine there are a lot of conditional probabilities and all that stuff that go into these estimations. And the guiding question here is how might disease prioritization impact wait time delays for patients in a hierarchical scenario? So rather than just prioritizing all AI positive patients the same, maybe you have a hierarchical disease priority where one type of stroke is prioritized over another type of stroke. And so we did some example simulation and theory validation with a brain CT workflow, assuming 2 disease conditions, 2 types of stroke, LVO and ICH. And over here, we're only assuming that there's one AI in clinic looking for LVO specifically. And so in this case, you can plot the wait time savings for true disease to LVO patients in the red line. And then the blue line are wait time delays for the ICH patients in this scenario. If you have 2 AIs, one looking for LVO and one looking for ICH, both patients are going to experience time savings. But if LVO is prioritized over ICH, as in this scenario, the time savings for LVO patients are going to be much larger, not much, but a bit larger than those for ICH. So just looking at these types of scenarios, and even if the FDA can't enforce exactly how AIs will be used in clinic, being able to publish papers and guidance documents that touch on some of these potential pitfalls of triaging scenarios, just to alert clinics, is something that we're interested in doing with this paper. So lastly, before I conclude, I just want to advertise some FDA opportunities. So in general, my division is hiring a lot of research fellows. And Aleem Thompson, who authored that software and has been my mentor this year and is great, is looking for two fellows in particular, because as I mentioned, I will probably be moving to some other projects. So there's still some work to do on rule out and some funding on that project available. And then there's another project kind of having to do in some sense with this CAD triage stuff, using a new-ish statistical methodology called generalized pairwise comparisons to be able to evaluate multiple endpoints simultaneously. So currently looking at diagnostic accuracy and time savings from CAD-T, people kind of cite those 2 metrics independently, but GPC would be a potential method to kind of incorporate all that stuff into the same analysis and kind of like a utility metric in some sense, but it's a lot cleaner than that. So those are 2 projects. There are also some projects involving multi-reader, multi-case studies and looking at variance estimates on those studies. And then everything you see up here, I mean, I went over some of the projects earlier as well. So to contact me if you're interested. And yeah, I think I'll end there. I have some extra stuff if people are like, but maybe we can talk about it after the. That'd be great, thanks.